Before we jump into any one step of urine formation, I just wanted to give you an overview of what urine formation is all about. It's actually very straightforward and it involves three steps. Uh, One step is filtration, and filtration is the exact type of filtration that we learned about in AP1, where we pass a solution through a filter being a semi-permeable membrane, something that has pores of a certain size. And if the particles are small enough to cross that filter, then they will. But if they're too big, then they won't. And so filtration in urine formation literally is that type of filtration. What happens is that a filter is formed between the glomerulus and the visceral layer of Bowman's capsule, which we'll talk much more about. But basically, there's a semi-permeable filter so that as blood passes through the glomerulus, any items that are small enough, small solutes and water, to get filtered out, physically leave and enter into the capsular space of Bowman's capsule. And anything that's too big doesn't, and it just leaves the blood through the efferent arteriole. So that brings me to my next question. Does that mean that all the small solutes that got filtered out of the blood are waste products? That everything large in the blood that didn't get filtered is fine? And all the small stuff that came out into Bowman's capsule are wastes? Hopefully you're thinking, that isn't the case. And it's not. It's very simplistic, right? Filtration is very simple. It's the separation of big from small. And so not all of the small solutes that got filtered out of the blood are waste products. And so actually the next two steps of urine formation have to do with correcting for this simplicity. So the second step in urine formation is reabsorption. So we said that small solutes and water are going to leave the blood, enter into the capsular space of Bowman's capsule, and then what happens is they start moving down the proximal convoluted tubule. So what we want to do next, particularly in the proximal convoluted tubule, because that's where the microvilli are located, is take some of the small solutes and the water that we don't want to get rid of, right? Because some of those solutes are nutrients like glucose and amino acids, various electrolytes and water. We don't want to get rid of all of that. And so what we do is we move it from the proximal convoluted tubule back into the blood of the peritubular capillary, and that step is known as reabsorption. So that brings me to my last question for this little video. What about the large substances in the blood that were too large to be filtered? Are any of those waste products? Is it possible for waste products to be large and they just didn't get filtered and so therefore they remain in the blood? And hopefully you're thinking, yes, that there can be waste products that are too large to be filtered. And so the third step of urine formation corrects for that. All right, so the third step of urine formation is called secretion, and that's when larger substances that remained in the blood just because of their size, they were too big to get filtered and yet they're waste products. What happens is they get directly secreted into the renal tubule. And so secretion is a process by which we take larger waste products and secrete them directly into the renal tubule. And so the net result after these three steps is the production of urine. So the production of urine involves the filtration of small items out of our blood into the nephron. We're going to take back through reabsorption the good things that we want to keep, like nutrients and water, and we're going to secrete the larger waste products that didn't get filtered to form urine. And so we'll look at each of those steps one by one.